back, advanced biology students. We are here today to talk about the exciting world of mitosis, and this is 8C. This is actually 8C this time. That's not a mistake. So we're going to talk about mitosis. But you may be asking, why do we care? Who cares about mitosis? Why should I care about mitosis? Well, mitosis is important to us as human beings. It's important to us as living creatures for a couple reasons. One, say, for instance, we're out and we get a nice big skin on our knee from doing something. You're out running, you trip, fall, you skin your knee. Well, mitosis is the process that's going to replace those cells. That's how we're going to repair the damaged tissue. That's how we're going to heal ourselves. So mitosis has this huge role for us in repair. But even more so than that, we start off our lives as a single cell. A single cell. When we're conceived, when egg meets sperm, we form a zygote, and that zygote is a single cell. And we go from a single cell to this huge, complex, multicellular baby, right? And then eventually this multicellular baby grows into a large, complex adult, and that process that we use to go from a single cell to this huge, complex adult organism is mitosis. So this is wildly important understanding for us human beings in our world, because this is how we got to be who we are. Okay, I want to start off first by talking about how we do this thing called maintaining the chromosome number. Because if you look at this, it looks kind of goofy, because we start off with one cell. So in mitosis, we start off with one cell with a certain number of chromosomes. In this case, we're starting off with two chromosomes, a red chromosome and a blue chromosome here. And at the end, we end up with two cells with two chromosomes. Well, that seems kind of goofy. How do we start with one cell with two, and then at the end we end up with two cells with two? If we're just splitting the cell, we should get half the number of chromosomes in here. Well, that doesn't happen because of something. In the cell cycle, in the S phase of the cell cycle, we replicate the DNA. So now we have two replicated chromosomes. So now we have two. There's still only two chromosomes. They're attached by these centromeres. We still only have two chromosomes, but they're replicated. Then what happens is these replicated chromosomes, they break up and split at the middle, and then we get two cells with two chromosomes. So we start with one cell with X number of chromosomes, and then we replicate them. So now we have X replicated chromosomes, and then we split them, and now we have two cells with the same number as we started. So that's how we maintain the chromosome number, is we duplicate the DNA. We replicate it during the S phase of the cell cycle. Okay? I want to make you familiar with the bits and pieces of mitosis before we get to how it all works, so you have an understanding of the parts and some key players that you may not know a lot about. And those key players I want to talk about are centrosomes and spindle fibers. Centrosomes are an interesting sort of contraption, for lack of a better word. And here's what they look like. Centrosomes are kind of these interesting contraptions here. They're made up of a couple of centrioles. Centrioles. So here's the centrosome. The centrosome is this whole sort of unit here, and it's made up of these centrioles, which are a type of microtubule. So they're a microtubule. And from that, we have a whole bunch of these fibers coming off, and they're called spindle fibers. Spindle fibers. And these spindle fibers attach to a region on a chromosome called a kinetic core. So they attach to this centromere at a spot on the centromere called a kinetic core. So spindle fibers are attaching to these kinetic cores, and they're also attached to these centrosomes. And these centrosomes, they have this job of controlling the spindle fibers. They're going to move these spindle fibers, contract them, relax them, and they're going to control them to get these chromosomes to line up in the middle, and then eventually to get them to split. So they're going to split the chromosomes right down the middle. So these centrosomes have an important role in sort of controlling the chromosomes. And if you think of a puppet, like a puppeteer controlling them, that's sort of what they do. They have these puppets attached to strings, and they can control them that way. Good way of thinking about it. Let's dive into the phases of mitosis. So mitosis takes place in a series of steps. In each of these steps, certain things happen, and we can identify what step they're in fairly easily by what the chromosomes look like, and we'll work our way through that in lab and become familiar with that from last year. But these are the phases that happen in mitosis. Now, mitosis is not a step-by-step. It doesn't like do prophase and then instantly warp to metaphase. It's a continual thing. So we may see cells in early prophase, or we may see cells in later prophase or middle prophase. So prophase can look a little different based upon how far along in prophase is it. And metaphase, anaphase can look a little different based upon how far apart those chromosomes have pulled. So we may see an anaphase that's right together, or we may see an anaphase that's far apart, because it's a continual process. So keep that in mind. The first phase is prophase. So it starts off with prophase. In prophase, here's what happens. The first thing happens is the chromatin. So the DNA is usually in the form of chromatin. But during this time, during prophase, the chromatin winds down into chromosomes. So now we're actually in chromosomes. And this makes them visible. You can see them with your eye or with a microscope. The other thing that happens is the nucleolus disappears. The nucleolus starts to disappear, and the nuclear envelope breaks down. So we're going to start to see this nucleus disappear. The other thing that's going to happen is these centrosomes are going to appear. See the centrosomes here? They've started to appear, and they're going to migrate. They're going to go to the poles of the cell. Okay, the next stage that happens after that is metaphase. Metaphase. In metaphase, what happens is these centrosomes, with their spindle fibers attached, are going to line the chromosomes up in the middle. So right here, they're lined up in the middle, and it's on an imaginary plane that we call the metaphase plate. It's not actually a plate. It's not a thing. That's just what we call it. So they're lined up on the metaphase plate, and that's metaphase. You can see the centrioles out here. They've moved to the poles. They're at either ends of the cell. They're out here. They move to the poles, and so we're lined up on that metaphase plate. That's metaphase. After metaphase comes anaphase. And in anaphase, what happens is the chromosomes break at the centromere, so they're going to split at that center. If we look at a chromosome, let's draw a little quick chromosome here. It looks like the Easter Bunny. Okay, they're going to split right along the center here. They're going to break here, and this part is going to go this way, and this part is going to go this way. So they're actually breaking at the centromeres. And these sister chromatids are going to move to opposite ends. So that's anaphase. So these things are all being moved this way, and these ones are all being moved to this pole towards the centromere. Once again, not centromere, the centrosomes. And once again, the centrosomes are the ones controlling the movement. They're using those spindle fibers to control the movement. Okay? Finally, telophase is the last phase of mitosis. And in telophase, we're starting to see this contractile ring that's actually closing or pinching off the cell here. This only happens in animal cells and plant cells. We'll talk about what happens with them. So we're getting this contractile ring here, contracting and pinching off the cell. We're getting the nucleus reforming along with the nucleolus. We can start to see that again. The chromosomes, they unwind. They go back to chromatin. They become invisible. And these centrosomes, they begin to disappear. So we're resetting. It's like the opposite of prophase. It goes back to where it came from. And at the end here, at the end of the whole thing, we're going to have two cells. Two cells with the same number of chromosomes as we started. And they're identical. They're clones of each other. Absolutely identical. Okay, that's telophase. Cytokinesis is this stage that happens. That's where the cytoplasm finally divides. So we're going to split this off, and when it finally divides, that's cytokinesis, or that dividing up of the cytoplasm. If we take a look at the results of mitosis, we'll just go through this quickly. We'll start here at this thing that's called the cycle start. They add in interphase and G1, part of the cell cycle. So if we look at G1, the cell's growing. This is in interphase in G1. The cell's growing. And then we get to that S phase of interphase, and we replicate the chromosomes. We can see now that these chromosomes are replicated chromosomes. After that, we go to G2, and the cell prepares to divide. It continues to grow, gap 2. Then we get into prophase, right? Everything starts to appear. Things start to break down. Those centrosomes appear and move to the poles. Then we go to metaphase. Everything lines up in the middle. Anaphase, they split. Telophase, they begin to pinch off and go back to their original form, where the nucleus reforms and the chromosomes wind down. And then eventually, we either go back and do it all over again, or the cell diverts into the G0 stage, in which it becomes inactive and doesn't divide anymore. So these two cells will either continue to grow, or they'll be going to G0 and become inactive. So that's the whole thing of mitosis. If you have any questions, class, feel free to ask me immediately so I can help you out. And yeah. Good luck. Good luck finding these in lab. And if you have any questions when we're in lab, let me know. Thanks. Have a good day.